Previously, we started looking at amino acids as the basic units of proteins. And we looked at 20 different amino acids which are found in proteins. We said there are other amino acids, except that these amino acids, you don't find them in proteins. You find them, some of them are intermediates in the metabolism of proteins. Some of them, even though they are having both the amine group and the carboxylic acid groups, they are different in structure compared to the amino acids which we find, these 20 amino acids which we find in proteins. They will be having similar functions, like acting like some neurotransmitters or some receptors on cells, but they are different in structure, and they are not found in proteins. We talked a bit about the naming of the amino acids. And so we said, for instance, asparagine. Asparagine is named as asparagine because it was first found, extracted from a plant. Asparagus is a vegetable plant which is found in Europe and Asia. Just like how we have spring onions, they have some plants of the sort asparagus, from which asparagine was extracted for the first time. Glutamate was also extracted the first time from the wheat protein, gluten. Gluten is the protein which is responsible for the elasticity of wheat dough, which is used in baking bread and other bakery products. It is that protein which is responsible for the rising or the bloating of the bread before it is baked. Tyrosine, coming from the Greek word tyros, which means cheese, because it was also first isolated from cheese, the milk product or the dairy product cheese. Glycine, also coming from the Greek word glykos, because it was found to be having a sweet taste. So, as I said earlier, yes, there are other amino acids apart from the 20 amino acids which are listed here, but those amino acids are not found in proteins. We looked at the structure of amino acids, having a central alpha carbon on which you have a carboxylic acid group, an amine group, and we have a side chain. There's also a hydrogen on that alpha carbon. So as I was saying, some of the amino acids which are not found in proteins, yes, they will have the amine group, they have carboxylic acid group, but not necessarily that this amine group and carboxylic acid group are found on the alpha carbon, meaning that there are some other carbons on which you find these carboxylic acid and amine groups, and they will be acting just like amino acids, but they are not found in proteins. Then we looked at the fact that the alpha carbon is a chiral carbon because it is having 4 different groups around it, 4 different groups on it. So you have an amine group, distinct, not like any other group which is on that alpha carbon. You have a side chain, which is also not like any other group. You have the carboxylic acid group, it's also different, and you have a hydrogen, and that makes the carbon a chiral carbon. But there is an exception, which is glycine. So because glycine, in place of the side chain of glycine is hydrogen, which means the alpha carbon will be having 2 hydrogen atoms on it. That makes the alpha carbon in glycine non-chiral. So for all the amino acids, glycine is an exception that does not have a chiral carbon. From there, we looked at enantiomers of amino acids. These are mirror images of each other. So you can have an L amino acid, D amino acid. These are stereoisomers in relation to the structure of glyceraldehyde, so that it's important in the functions of proteins, in the sense that as you go ahead to do biochemistry, you come to realize that these molecules act like switches, so that naturally you have amino acids that are existing in proteins in their L isomeric forms. So even though we consume some D amino acids, particularly from plant sources, these D amino acids in the metabolism of proteins get converted to L amino acid before there is further metabolism of those amino acids. So enantiomers, in some instances, will just be referred to as stereoisomers. How the molecules are, how the orientation of the molecule is in space. And so if you have an L isomer, you see the amine group is pointing to the left, whilst the hydrogen is pointing to the right. And you have a D isomer, where you have the amine group pointing to the right, whilst the hydrogen is pointing to the left. And this is how it's represented by what we refer to as the Fisher projections. These just lines, using just these cross lines to represent bonding, where the center of that cross is the alpha carbon. I think even back in senior secondary school, you may have looked at something like racemic mixtures, which is a mixture of two stereoisomeric forms in equal amounts, so that you in racemic mixtures, if you take a racemic mixture of say these amino acids, it means that you're having in equal amounts of these D and L enantiomers. So you can relate it to that earlier knowledge of racemic mixtures having equal amounts of the stereoisomers of a particular molecule. Then we looked at essential and non-essential amino acids, where we have essential amino acids being those amino acids that the body is not able to synthesize. The body is not able to make them. And so the body can only acquire this from food. So that is why it's being referred to as essential. You have non-essential being those amino acids that the body is able to synthesize them. So whether it takes it in from food or not, the body is able to synthesize these amino acids being referred to as non-essential. But there's not a thin line per se, because you have, for instance, arginine and histidine, which may be essential in childhood, but as we grow up, we are able to start synthesizing arginine and histidine. And And okay, I've just seen a message from the class rep that some students are not able to join with a link, so they want a meeting ID. They just have to copy paste in a browser. They don't necessarily have to even use an app, and they'll be able to access it. So they don't need any meeting ID and all of that. They should just tap on it and choose a browser and join. They shouldn't want to say they want to uh, install an app or anything of the sort. Okay. Now, in this sense, with essential and non-essential amino acids, the term complete proteins refers to a diet that contains both essential and non-essential amino acids. It's a term, say complete protein, it's a term relating to, say, balanced diet, for instance, so that you are having these two types of amino acids in there to tell that the food is whole and you have these two groups of amino acids being present, so that the body is able to synthesize the ones it's not able to synthesize, and the ones that is not able to synthesize too, it is available for it. Okay. We went ahead and started looking at the amino acids, all the 20 amino acids. We're looking at grouping of them in relation to their side chains. So we first looked at non-polar aliphatic side chains. So the amino acids have been such non-polar aliphatic side chains are what we see here: glycine, alanine, proline, valine, leucine, isoleucine, and methionine. Aliphatic only means these side chains are linear, except that you have proline, which is looking more like a side chain, which is looking more like a cyclic structure, but that's not a ring per se. It's just an internal bonding occurring between some internal interaction between the side chain and the amine group, which is giving us some form of a cyclic structure. But if you look at it, actually the side chain itself is linear. It's linear. The side chain does not have any cyclic structure in it. It's linear, but it's forming an internal bonding with the amine group. So we see glycine, the smallest of the amino acids, having hydrogen as the side chain. So intentionally, we have the side chains that are shaded. So that is what we pay attention to. So carboxylic acid group is having you have the amino acid having the carboxylic acid group on the alpha carbon, the amine group also on the alpha carbon. That is constant in all amino acids. So what we pay attention to is the side chain, which is shaded. So we have glycine having a side chain, which is hydrogen. So that is the smallest of all the amino acids. Then we have alanine, which is having a methyl group. And from then on, we see the side chains are getting more and more complex. So we have, from this alanine, you now have an increase of this one carbon there. You have 3. You have valine, also side chain being linear, leucine, isoleucine, methionine. So methionine, I mentioned that this is one of the 2 amino acids containing sulfur in proteins, methionine. And as you go ahead in biochemistry, maybe second year, proteins, so that in the synthesis of proteins in the body, the first amino acid that starts the process is always methionine. Then I also mentioned to you that when you read in biochemistry and you see any term like thio, THIO, or sometimes THIA, thia, it means that that molecule contains sulfur. And so you see, thioether group, the thioether group is the CH2S, CH3. That is the thioether group. And I showed to you that the ether group in chemistry is carbon bonding to oxygen, bonding to carbon. In this case, in place of oxygen, you have sulfur, and that makes it a thioether group. Right. We went ahead and looked at amino acids with aromatic side chains. So these are real cyclic groups, cyclic structures, such that you have phenylalanine, tyrosine, and tryptophan being the three amino acids with aromatic side chains. And these three amino acids, particularly tyrosine and tryptophan, are responsible for the absorbance of light of proteins around 280 nanometers. So we see the absorbance spectrum being shown here, and you see tyrosine and tryptophan shown here. So clearly you can see that tryptophan absorbs more light at that same wavelength than tyrosine, and that is logical because you have tryptophan having two rings and you have tyrosine having only one ring. You know, generally in chemistry, structures that have such aromatic side chains have this property of absorbing monochromatic light. So when you go to the lab to go and do some studies on proteins, along the line, you will do some protein extraction. And after the extraction of the protein, you do some analysis of the protein, which is you'll be using the spectrophotometer to measure absorbance of the protein. In that exercise, the absorbance you are measuring is actually due to the content of these amino acids, tyrosine and tryptophan. To a much lesser extent, even phenylalanine will have some absorbance. So a combination of these amino acids in the protein result in the absorbance of light of the protein at 280 nanometers. Even when you do extraction of nucleic acids, so DNA, and you are estimating the purity of the DNA which you have extracted, you use the same method of spectrophotometry where you measure the absorbance of your extract at 280 nanometers. And then you also measure the absorbance, I think, at a lower wavelength of about 260 nanometers or so, or to do a comparison. Because you have the nucleotides, which are in the nucleic acids, absorbing light at such a lower wavelength than that of the protein. So when you measure the absorbance at these two different wavelengths, you take a ratio of it. That ratio will tell you, so your extract is not purely DNA, but it contains some proteins in there as well. So it gives you a fair idea, estimation of how pure your extract is, how much of proteins are in there, and how much of nucleic acid is in there. So when you get to that practical, you remember this theory. That practical is based on this theory. Now we have Tyrosine having a hydroxyl group. That hydroxyl group is plays an important role in the functions of enzymes. So again, when you go much more into biochemistry, you come to realize that you have one form of uh, catalysis, or the function of the uh, increase in the rate of biochemical reactions in the body by enzymes. One of the mechanisms by which that occurs is acid-base catalysis. So that if you have a hydroxyl group here uh, in the side chain of tyrosine, this hydroxyl group can interact with protons or hydrogen ions, can be from another amino acid in the active center, the particular uh, portion, the pocket of the enzyme in which the reactions will be occurring, so that acid-base interaction between the hydroxyl group of tyrosine and a proton, a hydrogen, will result in the activation of the enzyme for the enzymatic reaction to proceed. We have looked at the polar but uncharged side chains, polar but uncharged side chains. So that these examples we see here, you have serine, and serine is also having a hydroxyl group in the side chain. The oxygen there is electronegative, and that results into polarity of that side chain, even though the side chain is uncharged. And you see the same thing, 309 is also having a hydroxyl group in there. So it will have a similar effect, just like serine. And cysteine, so this is the second amino acid having sulfur in it in proteins. So the first one we saw was methionine. Now we are looking at cysteine, which is having a sulfur hydroxyl group in it. And this sulfur hydroxyl group is also electronegative and will result into polarity of that side chain, even though it is uncharged. You see asparagine and glutamine, both of them are having some carbonyl group, a carbon with amine group and oxygen on it, a double bond O. So that group will also result into polarity of the molecule, seeing oxygen there and even an amine group. But these groups are not charged. So they are uncharged, but they are polar. Then we moved to look at positively charged or basic side chains. And here we see lysine is having an extra amine group apart from the amine group which is on the alpha carbon. You have an amine group in the side chain as well, and that amine group is positively charged. So that makes the side chain positively charged. Arginine is also having an amine group that is having a positive charge on it. The last one, which is histidine, here we are not seeing any positive charge per se, but remember we have here into bracket basic. So a base is will be receiving protons or hydrogen ions. So if you take an acid and a base, the acid gives out hydrogen ions. The base is the acceptor of the hydrogen ions. So if you look at the side chain of histidine, we can see that there are some double bonds that is showing some unsaturation of that structure. So that, for instance, this nitrogen that is having a double bond here with carbon, this nitrogen can accept a proton. So you can have a hydrogen coming onto this nitrogen and then a hydrogen coming onto this carbon as well. That is to remove this double bond from it. So this side chain is capable of accepting protons. That makes it basic. So even though it is not positively charged, it's basic because it can accept protons. Similarly, these two carbons that are having double bond between them can also accept protons. So there can be a hydrogen coming onto this these two carbons can have, each of them can accept protons on them, and that will make this side chain, the side chain of histidine, basic. We can't say it's positively charged, but it's basic in that sense. Indeed, histidine is also one of these amino acids which is found in the active center of enzymes, just like we saw with tyrosine having a hydroxyl group, and we said it was playing an active role in enzyme function. Similarly, histidine also plays. So this imidazole ring, we refer to this ring as imidazole ring in histidine. It also plays an important function in enzyme activity, in the acceptance of protons or hydrogen ions, which results in the activation of enzymes to go into an enzymatic reaction. Right. Then we have the negatively charged, which into bracket acidic side chains, and these two are aspartic acid. If you see aspartate is the same as aspartic acid, you see glutamate is the same as glutamic acid, and these two are having an extra carboxylic acid group in the side chain, and you see these side chains, in ionic form. Once they give out this carboxylic acid group that gives out hydrogen ions, and so it's carrying a negative charge in its ionic form. So it's acidic because it's a carboxylic acid group that gives out protons or hydrogen ions, and then itself in physiological conditions, in aqueous solutions, will be existing in its ionic form this way, carrying a negative charge. Right, so I think that was where we got to. That was where we ended previously. So we continue today from here, non-ionic and zeta ionic forms of amino acids. So we take amino acid. We can have the amino acid in non-ionic form. So the non-ionic form is having the alpha carbon, on which you have an amine group, NH2, and you have the carboxylic acid group. So you notice in this non-ionic form, or call it a molecular form, you do not find charges on these groups. So these groups are having just the amine group not protonated, not having an extra hydrogen on it. The carboxylic acid group too has not lost its proton, its hydrogen ion, and so it is intact. And so you find this uncharged, non-ionic form or molecular form of the amino acid. But we have the zeta ionic form. This is actually the form in which amino acids exist in aqueous solutions and in physiological conditions in the body. you have amino acids existing in their zwitter ionic forms. So then the word zwitter is coming from German, which means hermaphrodite. So this is just, and you know what a hermaphrodite is, an organism having both uh, male and female sex organs. In this case, we have uh, the amino acid form, the zwitter ionic form, having both positive and negative charge on it. Positive charge on the amine group and a negative charge on the carboxylic acid group. So that's the zwitter ionic form. Now, in this sense, you can have the zwitter ion, or the zwitter ionic form of amino acids acting both as acid and base. So in the first instance, you can have the zwitter ion giving out hydrogen ions from the amine group. So you see the zwitter ion giving out a hydrogen ion, a proton, and that NH3 plus becomes NH2. In that sense, it is acting as an acid, giving out a hydrogen ion. Now, the zwitter ion can also act as a proton acceptor or a base, so that you see the carboxylic acid group, COO minus, can accept a proton or hydrogen ion, so that now the COO minus, or the carboxylic acid ion there, is now accepting a hydrogen ion, a proton, to become the full protonated form, which is COOH. So same zwitter ion can act both as an acid, and it can also act as a base. So in this sense, amino acids in the zwitter ionic forms are referred to as ampholites or amphoteric electrolytes. So again, ampholites will be coming from the word amphibian. And we know an amphibian is an organism that lives in water and on land. So again, it's just referring to two cases, two instances. In this case, it is specifically referring to the fact that the zwitter ion is carrying both positive and negative charge. So these will be acting as some positive and negative charges at the same time, if you are referring to them as electrolytes, or if they are carrying electric charges, and these are amphoteric electrolytes. Now we see that you can have the amino acid, apart from, say, in its zwitter ionic form, it will be having a net charge of 0, because you're having a positive charge on the amine group and a negative charge on the carboxylic acid group. That makes the zwitter ionic form have a net charge of 0. But if you have a fully protonated amino acid, fully protonated, meaning that the amine group is fully engaged with protons or hydrogen ions, so that you now have a positive charge, NH3 plus. And you have the carboxylic acid group also fully protonated, so you now have COOH. That will be carrying no charge, but the positive charge on the amine group will now make the amino acid be carrying a positive charge, +1. In the other sense, if you have the zwitter ionic form giving out protons, that is acting as an acid. In that case, that hydrogen ion or the proton will be coming from the NH3+, the amine group. And that is going to result in a net charge of -1, which is on the carboxylic acid group, because now you have the amine group being NH2 carrying no charge, but the carboxylic acid group is carrying a negative charge. So that makes the amino acid carry a negative charge. So you can have amino acids in solutions having this dynamic form. Either it's carrying a net charge of 0, or it's carrying a net charge of +1, or it's carrying a net charge of -1, because the molecule is not static. It will be having interactions, and will be changing forms. Either it's having a form of the Zwitter ionic form, having a net charge of 0, or it is fully protonated and it's having a net charge of +1, or it has acted as an acid giving out hydrogen ions, and so will be carrying a net charge of -1. You can have these different forms of amino acids in solution. Now, we refer to Zwitter ions as being, or let's say, amino acids, we refer to them as being diprotic. So a Zwitter ion being diprotic in the sense that the Zwitter ion is capable of giving out, or let's say a fully protonated amino acid is capable of giving out 2 hydrogen ions or 2 protons. So this is what I mean. If you start from here, the left-hand side, where you are having a fully protonated amino acid, this fully protonated amino acid is having extra proton or hydrogen ion on nitrogen, and you also have the proton on the carboxylic acid group. So you see that you have 2 steps by which this fully protonated amino acid is giving out hydrogen ions or protons in each step. So the first step, you see the carboxylic acid group gives out a hydrogen ion proton and is now carrying a net charge of 0. Then you have the second step, where the NH3 plus is giving out a proton or hydrogen ion and now carries a negative charge. In that sense, you have a fully protonated amino acid which has given out 2 protons or 2 hydrogen ions. That makes amino acids diprotic acids. So 2 protons are given out. So you also come across the term diprotic acid. That is an amino acid, of course, having just one amine group. So it's having a mono amino group, and it's also having a mono carboxylic acid group. Such an amino acid is diprotic. Now, it's important to pay attention to the fact that it is only for a mono amino and a mono carboxylic, because we have seen some amino acids that are having extra amine group in the side chain, and we have also seen some that are having extra carboxylic acid group in the side chain. Those ones will not be diprotic, because you have those ones that are having an amine group in the side chain. That amine group will also be giving out a hydrogen ion or a proton. That will make it triprotic. Those having carboxylic acid group, which will also be protonated, can also be giving out hydrogen ions, and that will also make it triprotic. So it is only for amino acids that are monoamino and monocarboxylic, having this one amino acid group and one carboxylic acid group on the alpha carbon. These ones are what we refer to as diprotic acids when they are fully protonated. Right. So from there, we move on to a term also used in the study of amino acids, isoelectric point. Isoelectric point. So with what we have seen so far, that you have amino acids that can exist in different forms. In the non-ionic form or molecular form, and then they can also exist in zeta ionic forms. Then, apart from the zeta ionic forms, we see that they can exist in forms having a net charge of +1. That is when the amino acid is fully protonated. Or they can exist in a form having a net charge of -1 when the amino acid is fully deprotonated. So if we look at amino acids in the sense of having acid-base interactions in solutions, it means that, so if we take glycine as the smallest amino acid to model this, take glycine. So this is glycine, fully protonated. And first step, it gives out a proton or a hydrogen ion from the carboxylic acid group. You now have the zeta ionic form. And then the second step, it gives out proton or hydrogen ions from the amine group. And so you now have that amine group being NH2, and it's now carrying a negative charge. If in a solution, we are looking at this donation of protons from the amino acid glycine as an acid, what we observe is that at a certain point, at a certain pH, where you have the glycine molecule, the amino acid, having a net charge of 0, meaning it's now in its zeta ionic form. So remember that the solution of, say, glycine is not just one molecule of glycine, but there are more molecules of glycine which are dissolved in the solvent, water. At a point where all of the molecules of glycine are in their zeta ionic forms, it means that that solution is going to be having a net charge of 0. And that will occur at a particular pH. The pH at which the net electric charge of the solution is 0 is what we refer to as the isoelectric point. Now, it's not going to be the same for all amino acids, because the ability of an amino acid to give out its protons are not the same. So for instance, if you take glycine, when it is fully protonated, you'll find here the first value here, pK1 is 2.34. And the pK is a measurement of the ability of an acid to give out hydrogen ions. And you see the pK2 is 9.6. Actually, the lower the value is, the stronger the acid, the easier it is for the acid to give out hydrogen ions or to dissociate in solution, so that it is easier for the fully protonated amino acid, glycine, to give out a hydrogen ion or a proton than it is for the Zwitter ionic form. So that you have these two values not being the same. The first value is lower, showing that it's easier for the fully protonated to give out hydrogen ions. But it is at this point that the isoelectric point, it is at this point that you have the fully protonated amino acid that has finished giving out its first proton and is just about, starting to give out the second proton. So that is a midpoint, is a middle point in that interaction of donation of protons from the amino acid. So you keep that isoelectric point, that is the pH at which the net electric charge of a protein solution will be having a net charge of 0, the pH at which the solution of a proton, the solution of a protein will be having a net charge of 0. That is what we refer to as the isoelectric point. So if you have a solution of protein solution, and at a particular pH, the net charge of that solution is 0. What it tells you is that at that point, roughly, in estimation, you may not have exactly 0, but may close to 0. At that particular point, all the amino acids that are present in solution have assumed or are in their Zwitter ionic forms. That's what it means. So you have a solution that you have all the amino acids in their Zwitter ionic forms. The net charge is going to be 0, and that is the isoelectric point at a particular pH that is occurring. That is the isoelectric point, sometimes also referred to as isoelectric pH. Right. Now we have been talking about amino acids. So let's move on. Let's scale up. From amino acids, we start to form proteins. And in the formation of proteins, there must be bonding between amino acids, which are the basic units of proteins. So we see if this is one amino acid with a carboxylic acid group having an orientation close to the amine group of an adjacent amino acid, an amino acid close to it, you can have interaction between these OH, the hydroxyl group of the carboxylic acid group of this first amino acid, and this hydrogen, also in red, of the amine group of the second amino acid. Now, take note that the interaction which is occurring between this hydroxyl group and this hydrogen is not an acid-base interaction. It is not an acid-base interaction because, remember, this is a carboxylic acid group. And so if it's a carboxylic acid group, if it was going to be an acid-base interaction, it should be interacting with only the hydrogen. The hydrogen it gives out, that is the proton, as we saw earlier with the amino acids. It gives out hydrogen or a proton, H plus, and it's left with COO minus. But in this case, it is this whole hydroxyl group which is interacting, reacting. So it is not interacting as an acid. So this interaction we refer to as a condensation reaction. In some instances, we refer to it as dehydration, removal of water. So it's a condensation reaction. Condensation too is coming. I mean, if you liken it to meteorology, when it's coming to rain, being referred to as condensation. So you have this hydroxyl group, this hydrogen from the amine group interacting to give out water, and the result of it is the formation of the amine group, which is shaded in yellow. So you have actually the amine group, the peptide bond is between the carbon from which the hydroxyl group is gone and the nitrogen of the amine group from which the hydrogen is gone. You have the peptide bond between that carbon and that nitrogen. And you see the whole group which is shaded yellow, that becomes an amide group. So an amine group reacts and forms a bond and now forms an amide group. Now we have what we have here now, the product of this reaction is a dipeptide because it contains 2 amino acids. If you have a third amino acid attaching either at the N terminus, which is the amine group end, or the C terminus, which is the carboxylic acid end. If you have one amino acid coming to attach to that, you have a tripeptide. You can have tetrapeptide, that is having 4 amino acids. You can have pentapeptides, hexapeptides, heptapeptides, etc. Usually, from a dipeptide up to about 20 amino acids, these are referred to as oligopeptides. And then polypeptides will be those protein chains that are having more than 20 amino acids in them. So you can have very large molecules of proteins having so many amino acids in them. Those we refer to as polypeptides. Now, we can move on again, another level. So we just looked at the bonding that occurs between amino acids to form a dipeptide, tripeptide, or oligopeptides, polypeptides. In this sense, we have 4 different levels of protein structure, 4 different levels. The first is the primary structure of proteins, which refers to the sequence of amino acids in a peptide chain. So, for instance, if you are talking about the amino acids that are found in a particular protein that, oh, this amino acid contains lysine, which is bonded to another lysine, which is bonded to glycine, which is bonded to another glycine, bonded to leucine, bonded to valine, bonded to alanine, bonded to histidine. What you are referring to is the primary structure of that protein. You are only referring to the sequence of amino acids in the peptide chain. Then you have a secondary structure. That secondary structure will be formed from. So from this primary structure, you can have the protein beginning to form a ribbon-like structure with some hydroxyl groups that are holding carboxylic acid side to hydrogens of the amine groups of other adjacent amino acids, forming some ribbon-like structure, which we refer to as the alpha helix. We can also have what we refer to as beta sheets. These are secondary structures. If you have a protein in which you have two or more secondary structures, that is a tertiary structure. And if you have a protein in which you have two or more tertiary structures in it, we refer to that as quaternary structure, which you find in proteins like hemoglobin, for instance, that is having 4 different subunits acting as one. For tertiary structure, you may look at, say, myoglobin, which is found in the skeletal muscles, responsible for the transfer of oxygen. So it serves as an intermediary between hemoglobin and the muscle cells. So it will take the oxygen from hemoglobin and transfer it to the muscles. That is having only one globule, one protein which is having a tertiary structure, different secondary structures in there. Now I know how these can be sounding very abstract. So what we'll do now is that we will look at a video of this. We'll look at a video of this so that we have a better understanding of what we are talking about. So I will have to stop sharing this first, okay? And then we look at, okay, so just a moment, let me bring the video up, and then it's an animation so that clearly we can see the various levels of protein structure that we are referring to, which makes it much clearer than if we are just talking of it on the slide. Okay, so I hope now that can be seen. So this is two amino acids, alanine and glycine. Now, we see here, so the blue you see on your screen, that is nitrogen. The whites are the hydrogens that are bonded on that nitrogen. The black is carbon. The red is oxygen. So that black double bond O and then red OH, that gives you the carboxylic acid group. The black with the 3 whites on it there for alanine, that is the CH3 group. So black balls are carbons. Blue, the balls that are blue, these are nitrogen, and the red ones are oxygen. So we see two amino acids, alanine and glycine, coming together to have such an interaction to form a peptide bond. So we see that. So we see that an OH group moves away from one of these amino acids and is going towards the hydrogen of the amine group of another amino acid. Look at that. Water is formed and water moves out. And then you see a bonding between the carbon and the nitrogen of these two amino acids. So that is a dipeptide. That's a dipeptide, having just two amino acids. Now we see this reaction repeats. So that I have cysteine asparagine also coming. And look at that. Water is formed, goes out. Bonding between these. You now have a tetrapeptide. Now have a tetrapeptide. So this is a protein containing 4 amino acids. Now this can continue on and on and on and on, and you have a long chain peptide. Now you have a primary structure, which is just a sequence of the amino acids in the peptide chain. Now, from that primary structure, you can have a ribbon-like structure forming where you have hydrogen bonds forming between some hydrogens and oxygens. So oxygens of the carboxylic acid groups and the hydrogens from the amine groups. So you have a secondary structure, which is alpha helix. You can also have the peptide chain folding such that you have these parallel structure. You have the peptide chains having this parallel structure such that you now have hydrogen bonds between carboxylic acid groups and hydrogens of amine groups. That is beta sheets. So you have secondary structures being the alpha helix and what we refer to as the beta sheets or the beta pleated sheets. Now you are having different alpha helixes that come together to give you the structure of a protein. That is a tertiary structure. So you have more than one tertiary structure, more than one secondary structure coming together to give you a tertiary structure. Similarly, you have more than one tertiary structure coming together to give you a quaternary structure, as we see in the molecule of hemoglobin. So that is what we were referring to. I will share not you already have the presentation. I will also share the animation which we have just seen. I will share it through the class web so you can watch it multiple times to have a better understanding of what we are talking about. Right. So what we just saw in that animation, the first was the primary structure, which we talked about already as the sequence of amino acids in a peptide chain. The secondary structure, you can have the alpha helix, where you have the oxygen of carboxylic acid groups and hydrogens of amine groups having some hydrogen bonding interactions between them. and that forms some ribbon-like structure. So we see these bonds being shown here in dots at the right side of the slide you are looking at. So you have this as a secondary structure, the alpha helix as a secondary structure. We also just saw the secondary structure, which is beta sheets, where we have the proton folding to have the peptide being in parallel. So you have some parallel and anti-parallel structures, and you are having hydrogen bond interactions between the oxygen of a carboxylic acid group, which is close to the hydrogen of maybe the hydrogen on the alpha carbon. You see that you have such hydrogen bonding on and on throughout in such parallel and anti-parallel structure, which is having this some zigzag shape that we saw in that animation that we refer to as the beta sheets, sometimes being referred to as beta pleated sheets. Then we talked about the tertiary structure, which is having more than one secondary structure in it. So we see the sides which are, the ribbon-like structure, that's the alpha helix. So we have this alpha helix, which is having interaction between another alpha helix, and this interaction actually is occurring between the amine group, along the line in one of these alpha helices, and the carboxylic acid group from the other alpha helix. Such an interaction we refer to as a salt bridge. We can also have hydrogen bonding interactions. So you remember that in the secondary structure, what you have there has been hydrogen bonds. But now in the tertiary structure, apart from hydrogen bonds, you are having other interactions. So you have the salt bridges. You can have hydrogen bonds also there, where here this will be the side chain of serine, CH2, and you see the oxygen of that hydroxyl group of serine is having interaction with the hydrogen of another hydroxyl group of another serine side chain. So that is hydrogen bonding between oxygen and hydrogen occurring between two different. So this is coming from an alpha helix, and this is also is coming from just a straight peptide chain, because out of the alpha helix, almost like just along a certain loop, you have that hydroxyl group from the side chain of serine having such an interaction. Then you have, so here you can relate to it as the beta sheets, where you have peptide chains that are arranged parallel to each other, and you are having such hydrogen bonding between the hydrogen of the alpha carbon and the oxygen of a carboxylic acid group of another amino acid in that protein, forming these hydrogen bonds. You can also have interactions that we refer to as disulfide bonds. So the disulfide bonds will be existing between two cysteine molecules. So remember, cysteine is having a side chain with a sulfur hydride group, SH. And so you can have the two sulfurs having such an interaction between them, such bonding, which we refer to as the disulfide bonds. You can also have hydrophobic interactions between hydrophobic side chains, like the interaction between two phenylalanine groups. So these are two phenylalanine side chains that are having hydrophobic interactions between them, or even two alanine. So alanine is having a side chain of CH3. The two of them, they are hydrophobic, and they can have such hydrophobic interactions. And even the protein in aqueous solution, portions of it, like if this is tyrosine side chain, this tyrosine side chain is having some interaction with water molecule there. And you have even serine, which is having a side chain, CH2OH, also having some interaction with water molecules. So this is to show the various bonds that can occur in a tertiary structure. You have the salt bridge, aside the hydrogen bonding. You also have disulfide bonds, and you have hydrophobic interactions that are responsible for the tertiary structure of proteins. So keep those in mind. The hydrogen bonding, 1, salt bridge, 2, disulfide bonds, 3, hydrophobic interactions, 4. These 4 bonds or interactions are responsible for holding a protein structure together in a tertiary structure. And it is this tertiary structure that if you have more than one tertiary structure coming together, like in the molecule of hemoglobin, where you have 4 subunits, each of them is having a tertiary structure that is coming together, you now have a quaternary structure. So which means that the same bondings, the same bonds which you saw in the tertiary structure, it is those same bonds that you are going to find also in the quaternary structure. So here we are seeing from primary structure, which is a sequence of amino acids in a peptide chain, forming a secondary structure, which is ribbon-like, having hydroxyl groups between the hydrogen of the alpha carbon and the oxygen of the carboxylic acid group. Then you have you can also have, so here we are not seeing the second type of secondary structure, which is the beta sheets or the beta plated sheets, which will be having a hydrogen bonding between parallel chains of proteins or parallel chains of peptides having such hydrogen bonding between them. That is another type. So we have 2 types of the secondary structures, the alpha helix and the beta sheets. And then that from there we have the tertiary structure, which will be having more than one secondary structures in them. Here we see these are shown as alpha helixes. So different alpha helixes that are together. So sort of a protein folded to be functional. And then you have different tertiary structures coming together to now give us a quaternary structure. That's what we see here. Now we can talk about denaturation of proteins, which occurs with the action of temperature, high temperature, heat, acid, so some acid conditions, very low acid conditions, or very high pH, which is a basic condition, or even some heavy metal salts, or just agitation can cause denaturation of proteins. Now, what is denaturation of proteins? So the denaturation of proteins is the destruction of the structure of proteins such that the protein loses its functionality. So remember that denaturation of proteins does not result into the breakdown of protein into individual amino acids. No. The breakdown of proteins into individual amino acids occurs with metabolism metabolism, particularly with catabolism, the breakdown into various amino acids, which will now be used for synthesis of proteins. But in this case, you only have the protein which is having, say, a quaternary structure or a tertiary structure or a secondary structure. It is those structures that are destroyed. So the structure, the tertiary structure, quaternary structure, secondary structure is destroyed, but you still have the primary structure intact. The sequence will still be there. The protein will still have that sequence of amino acids. But remember that the primary structure of proteins does not give functionality to that protein. What gives functionality to the protein is its secondary, tertiary, and quaternary structure, because it is then that the protein is having a particular form. It folds in a certain way and starts to have a certain activity. So we see it being depicted, for instance, for X. So if it is agitation, just by beating the egg, you see you would have destroyed the structure of the proteins that are in the egg yolk. And if you apply heat again, that will cause also further denaturation of the protein. Now we see, so you see, when you are talking about digestion, for instance, digestion, you see that in the mouth, you have some enzymes that are acting. So the salivary amylase is breaking down carbohydrates. And in the mouth, the condition there is, slightly basically something around almost neutral, because it hovers around 6 points and 7 points. And you have the salivary amylase acting there. But in the stomach, you have hydrochloric acid, which means you have an acidic condition in the stomach. And different enzymes will be acting there. So you see, you have enzymes which are proteins being active at different pH conditions. So depending on the particular place where an enzyme is acting, a particular pH will be optimum, will be the condition for that protein to be active. So in other words, the salivary amylase will lose its activity in the stomach, because there the condition is different from the mouth. So the same way, those enzymes that are acting on proteins in the stomach will not be active in the mouth. Some metals, heavy metals, these are they have the functionality of forming complexes with proteins. So they refer to as chelation. And these heavy metals, when they bind to proteins, they can turn these proteins inactive. So you can have when you go to the lab, you have one of the common chelation, the chelation reagents you use there is EDTA. But apart from that, you realize that there are heavy metals like nickel, cobalt, copper, which can interact with proteins. And some of them actually serve as cofactors. These are needed for the activity of the enzymes. So if they are not there, the enzyme will not be active. So they can also affect the structure of proteins, resulting into denaturation. Now, we can broadly look at the functions of proteins, that proteins can be acting as enzymes. And these are biological catalysts. They speed up biological reactions. And these will go into a separate field of biochemistry referred to as enzymology. That is a whole course that you can go read enzymology at your master's level, PhD, just enzymology, for instance. So you have structural proteins that are forming body tissues, muscles, hair, nails, like keratin, all of that. That's studying structures of tissues, muscles, and things like that. We'll go into histology. You have some proteins which are they are called either transporters, membrane proteins. They are embedded in proteins, or some are channels which are responsible for the transport of ions across cell membranes and the transport of substances across membranes in and out. That's another function of proteins. If you are doing cell biology, or even if you specifically you say you are studying membranology, where you are studying biomembranes or cell membranes, you come across these types of proteins. Hemoglobin, a common protein you have known for the transport of oxygen in blood. So hemoglobin takes in oxygen from the lungs and takes it to the various tissues where oxygen is released to the cells to be used for cellular metabolism. And part of the carbon dioxide, which is the byproduct of cellular metabolism, is carried by the hemoglobin back to the lungs to be All right, I hope you can hear me now. I just got my lights going off and the internet cut. So, but there's some backup internet here, so I hope you can hear me now. If you can hear me, let me see my hands, so I'll be sure that I'm back online and you can hear me. Okay, great. So, I mean, we are even almost done, so we will just quickly finish up, and then I think we've been lucky this time around. This light has not disturbed us from the beginning. So we have just talking about the very last things, what proteins, their various functions. You can have proteins acting as neurotransmitters, where these proteins transmit signals in the nervous system, so signal transduction or communication between cells. You have these neurotransmitters acting in the nervous system along neurons, and you have some proteins also acting for immunological defense. Actually, white blood cells, you can say they are proteins because, as the name suggests, immunoglobulins. So if you see globulins, that is a protein. A globulin is a protein. Immunoglobulin is only mean a protein, an immune protein, or a protein having immune function. So you have proteins also having such a function for immunity. Some of the proteins are also hormones. We have some peptide hormones, which carry out some regulatory function in the body. These are also some form of messengers. So you see, if you compare neurotransmitters to hormones, neurotransmitters will be transferring electric signals along the nervous system. It's a way of communication in the body. But hormones, these are chemicals that actually move from one end to the other to cause an activation. So they are not transmitting a signal by electric charges, but they themselves, the hormones themselves, can move. Either they move from one place to the other, or they attach to a receptor and activate that receptor, which will cause other reactions to occur. So cascade of reactions will be occurring because that hormone is attached to a particular receptor. So receptors are also, they can be proteins that form complexes with carbohydrates, what we refer to as glycoproteins on cell membranes. And they are responsible for cell recognition. They can recognize substances and have some specific interactions with them, which will result into some specific responses. So you have proteins having several of these functions. And the study of proteins alone can give you several fields of science, or even particularly biochemistry, enzymology, or histology, neuroscience, immunology. If it's hormones, then you might be referring to endocrinology, for instance. If you studying just maybe receptors and transporters, membranes, that will be some cytology or cell biology. So I hope this is understood. So we have come to the end of this today. Let me just quickly look at the messages that people were writing. Somebody was saying he couldn't hear. I hope that later the person could hear. Somebody said, check your phone. Well, I think I saw the message. Then, okay, somebody asked, please, is it acceptable to write the amine group as H2N? So I think what you are referring to is, sometimes when you write the carbon, the alpha carbon, and you are writing the amine group on the left-hand side, you want to show that it is the nitrogen which is bonded to the carbon. And so you write that the hydrogens come before the N. So you say H2N. Yes, it's understood. It's understood. Except if you write the amine group on the right-hand side, and you write H2N, then it looks as if it is the hydrogen which is bonded to the carbon, two hydrogens, and then the nitrogen is also bonded. I think this one, when we meet in class, the person who asked this question, you can remind me so that I can explain it on the board further, so that you can you have an understanding of what I'm saying. So yes, H2N, yes, it's it will be clear in some instances, but in some other instances, it will not be clear. It's a language. It's a universal language. It's a chemistry. So if you are writing it, it doesn't have to leave any ambiguity. When somebody is seeing it from another place, uh, anywhere in the world, the person should be able to understand what you are writing. Somebody said I should repeat it. I don't know what exactly he said I should repeat. Somebody says my sound is muted, which makes it difficult to hear. Well, which means it was your setting. So you should have been able to. Okay, somebody wrote you that you should choose speaker. And then somebody said, okay, are peptide bonds hydrogen bonds? No, peptide bonds are not hydrogen bonds. So a peptide bond, remember, is formed between the carbon of the carboxylic acid group of one amino acid and the nitrogen of the amine group of another amino acid. That is a peptide bond. A hydrogen bond is a weak bond which is formed between hydrogen and oxygen. So you saw the hydrogen bonds that we were looking at were formed between, you can have a hydrogen bond between the oxygen of water with hydrogen of an amine group of an amino acid, etc., or the hydrogen of the hydrogen which is on the alpha carbon of an amino acid with the oxygen of the carboxylic acid group of another amino acid. So you see that. Now somebody also says, I don't know what exactly the person is referring to. He says, what is the name of the color yellow element? I don't know exactly the color yellow element. I don't know if the person can clarify color yellow element. I don't know exactly color yellow element. Maybe when we were talking about the bonding, maybe this, maybe the hydrogen, I think maybe this one, the yellow, the amide group, the amide group. Maybe that's what the person is talking about. After you have an amine group forming a bond, you now have an amide group there. Okay, so some people's hands are up. Is it when I ask the question that if you can hear me, raise your hand, or it is that you have a question? So if you don't have any question to ask, kindly put your hands down. Then if you have a question, your hand is up. I will allow you to. I will allow you to unmute yourself. You speak, and then respond. Teresa, Juby, do you have a question? Okay, Dorcas. Okay, so you, I will just come in. Let me allow you to